A key challenge in modern genomics is that more than 98% of human genetic variation occurs in non-coding regions. These so-called dark matter areas of the genome don't build proteins directly. Instead, they act as a complex regulatory switchboard. For clinicians, this creates a massive interpretation bottleneck. When a patient presents with a variant of uncertain significance in a non-coding region, determining whether it disrupts a distal enhancer, alters a splice site, or changes chromatin accessibility has been historically quite intractable without extensive lab work. The vision behind AlphaGenome, recently published in Nature, is to provide a unified computational framework that can predict these diverse molecular consequences directly from the DNA sequence. It aims to replace a fragmented ecosystem of specialised models with a single high-resolution kind of oracle for the regulatory genome. The primary hurdle between this conceptual breakthrough and clinical utility is the historical trade-off that there's been between vision and detail. Previous models could either look at long stretches of DNA and kind of visualize a lot to find things like distal enhancers, but at a blurry resolution, often at least 128 base pair bins. Or they could look at single base pair detail, but only ever look at short distances. This meant that a model might see a promoter but miss the enhancer 100 kilobases away that actually controls it. AlphaGenome attempts to solve this by processing a massive 1 megabase sequence window while maintaining single base pair resolution. The architecture is something called a UNet style design. It uses an encoder to downsample the sequence and transformer blocks to model long-range interactions, such as enhancer-promoter loops. Importantly, it leverages sequence parallelism across 8 interconnected devices to handle the computational load of 1 megabase inputs at base pair resolution. This training involved a 2-stage process. First, pre-training on experimental data across 11 modalities, including RNA sequence, cap analysis of gene expression, and chromatin immunoprecipitation sequencing. This was then followed by a distillation phase. In distillation, a kind of student model learns from an ensemble of teacher models using mutationally perturbed sequences. This makes the final model more robust at predicting the effect of rare variants and significantly faster, requiring less than 1 second of a modern GPU processing unit. What this enables is a comprehensive multimodal view of a variant. In the study, the model was evaluated on 26 different variant effect prediction tasks, achieving state-of-the-art performance in 25 of them. It particularly excels in predicting splice junctions, the specific points where introns are removed from the genome, which is a step beyond just predicting splice sites. It also showed a 25% improvement over previous models in predicting the direction of effect for expression quantitative trait loci, or eQTLs. This means it can more accurately forecast whether a mutation will increase or decrease gene expression, a vital piece of information for interpreting GWAS results. However, the path to the clinic remains gated by significant limitations. While AlphaGenome is still highly accurate at predicting molecular tracks, the raw data of the genome, it doesn't directly predict clinical phenotypes. There's still a kind of phenotype gap. Knowing that a variant reduces TAL1 oncogene expression in CD34 positive cells is a molecular insight, but not yet a clinical diagnosis. Furthermore, the model still struggles with extremely distal interactions beyond 100 kilobases and cannot yet accurately predict personal genome variations, the subtle interactions between multiple variants in a single individual. So it's a really interesting development, and it would be particularly interesting to see this progress to some sort of prospective validation in a diagnostic setting, specifically focusing on its ability to resolve variants of uncertain significance in rare disease cohorts. And you can envisage a future where implementation that integrates AlphaGenome scores into existing clinical annotation pipelines could provide a kind of functional evidence tier rather than as a standalone diagnostic tool. So AlphaGenome represents a shift from specialised narrow genomic models to a generalist foundation. It provides the most detailed map to date of how sequence changes ripple through the regulatory machinery of the cell. For the clinician, it offers a way to move from "we don't know what the variant does" to "this variant is predicted to disrupt the MIB binding motif and reduce target gene expression by 40%. But it doesn't yet bridge the gap to disease on its own, but it provides one of the most precise tools that we've ever had to start building that bridge.